Good evening, everyone. My name is Enda Guinan. I'm the Digital Communications Manager at Sarcoma UK. You're extremely welcome to this, which is the third in our series of Zoom chats about all matters sarcoma this July Sarcoma Awareness Month. We've already had sessions about sarcoma and women's health and clinical trials and sarcoma, and both of those are available at sarcoma.org.uk/Zoom. And like every other link we mentioned tonight, we'll pop that into the chat so you can find it later. So we are honoured this afternoon or this evening to have a panel of some of the, I don't want to exaggerate, but some of the most important people working in genomics and sarcoma today. It should be a fascinating 45-minute session looking at a very exciting and perhaps a little complex subject. And because it is a little complicated, we really do encourage you to ask questions tonight. And I'll talk about how you can do that in a moment. So if you're thinking of something, chances are other people are thinking of that too. So please feel free to ask questions. A couple of bits of housekeeping before we get going. The webinar is being recorded, so it will be shared on the website tomorrow, so you can refer people to it. Your details won't be on it, so don't worry about that. Some of you will have taken part in Zoom meetings before, and some of you will have taken part in webinars. The basic things to know are that we, the hosts and the panelists, we can't see or hear you. So you can enjoy your anonymity for the next 45 minutes. But if you would like to ask a question, there's a couple of ways to do so. There's a raise your hand box at the bottom of the screen there, and if you press that, I'll see it and I can spotlight you. So that means then you can ask a question, but you will be seen and heard by everybody. If you feel a bit shy about that, you can also type your question in the chat box. Again, everybody sees that. Or you can type it in the Q&A box and only the panelists see that. So again, we're encouraging you to ask your questions tonight as we go along. But first, I want to tell you a little bit about this stellar panel that we have for you. Our first panelist is Andrew Beggs, and Professor Beggs is the founder of the Beggs Lab, and he's a reader in cancer genetics and surgery at the Institute of Cancer and Genomic Sciences at the University of Birmingham. He's a member of the Human Cancer Models Initiative and a member of the consortium that sequenced the reference human genome. Professor Beggs is also a member of Sarcoma UK's Research Advisory Committee. Richard Davidson is the Chief Executive of Sarcoma UK, only the second person to hold that post in our 10 years. In fact, this year marks our 10th anniversary, and you can see what we have lined up at sarcoma.org.uk/10 years. And that's TEN. Richard came to us from Anthony Nolan, where he was Director of Engagement, and he's also the Chair of SameYou, which is a brain injury and stroke charity founded by Game of Thrones star Emilia Clarke. Adrian Flanagan is Professor of Musculoskeletal Pathology at University College London's Cancer Institute and one of the most distinguished academics in her field. And indeed, she was awarded an OBE in 2017 for her services to cancer research. Professor Flanagan is the lead on GSIP, Sarcoma Genomics England Clinical Interpretation Partnership, which is an ambitious UK-wide study of the 1,000 genomes taken from sarcoma patients and their families with the aim of developing personalised medicine for patients with sarcoma, and I'm sure she's going to tell us a little bit about that later. Then we have Dr. Alexander Lee, who is a consultant medical oncologist at the Christie in Manchester. Dr. Lee's general medical training was in Manchester and London, and his specialist oncology training was at the Royal Marsden. His PhD study carried out within the sarcoma unit at the Marsden and the molecular and systems oncology team at the Institute of Cancer Research before he was appointed to the Christie as a consultant last year. And we're interested in hearing about his work tonight too. Finally, we have Dr. Zoe Walters. And Dr. Walters is currently leading a study on genomic data and liposarcoma at the University of Southampton, where she is lecturer in translational epigenomics and module lead on the Masters in Genomics within the Faculty of Medicine there. In 2015, Zoe was awarded a Children with Cancer UK fellowship to study the role of an epigenetic protein in rhabdomyosarcoma, part of her research focused on finding more effective, less toxic treatments and novel therapeutic strategies for pediatric disorders. So as you can see, it is a panel of heavy hitters tonight. So please, if you have a question about genomics, these are the people who will be able to answer it. So right now, I'm going to hand over to my colleague Bradley, and he's going to take you through the rest of the evening.
Thank you very much, Enda, and a big welcome from me as well. I'm Bradley. I'm the Policy and Public Affairs Manager at Sarcoma UK. And I'm going to kick off by passing over to Andy and asking the very simple question, what is genomics and why is it so important?
Thanks, Bradley. Genomics is the easiest way to sum it up. It's understanding the genetic code of your cancer and of yourself, to understand what's gone wrong in the cells, to understand how we can better select treatments and therapies and work out what your prognosis will be in cancer. And it's not just about the code, it's about what the code turns into, something called RNA, and it's about modifications to the code called epigenetics as well. And altogether, these changes give us an idea of how we can better treat and understand cancers and what their prognosis are in the longer term.
Fantastic, thank you. And so what excites you most about genomics for sarcoma at the moment? Just a brief kickoff.
I think the most exciting thing about genomics and sarcoma is that there are so many diverse types of sarcoma at the moment, and they're all quite different in how their genetic code changes and is different. And I think the exciting thing is it's very much, there are some sarcomas where we know reasonably well what's going on, but there's very much an undiscovered landscape with some of the rarer ones. And from that, hopefully we could develop new treatments. The tools and the technologies we've got, they're the most exciting they've been in 15 or 20 years. And through that, we can hopefully transform the way we treat it.
Thanks. So moving over to Adrienne, setting the scene a bit further, could you tell us a bit about the 100,000 Genomes project and why sarcoma was involved with that?
So the 100,000 Genome Project was really a monumental study set up by the Cameron government in 2016. It ran for 5 years. It was extraordinarily ambitious and maybe somewhat naive in terms of what the challenges that lay ahead. But I think people realize there is so much information that can be got from patients' tissue, just one small amount of their tissue, that is all held, as Andy was saying, in the genome. And by pulling all of that together, we can, if we correlate that, and this was the ambition of the project, with clinical outcome, with then using that data for things like developing new tests to measure, could we identify markers, liquid biopsies, so we could monitor patients without having to biopsy them again? Could we use algorithms or artificial intelligence to correlate what's in the histology with the genomic data? And so this 100,000 Genomes project is an enormous resource that will last for the next 50 years, on which we're going to build slowly but methodically, and none of it's going to be wasted. But we will have benefits coming through step by step. We don't have to wait for the 20 years, but the resource is going to be absolutely phenomenal in terms of understanding the disease, using that information for developing new tests, for giving patient information, for developing new therapies. And there's so much information that doctors and healthcare workers can no longer hold it all in their brain. It has to be held by computers, which we can then dip into and analyze. And I think that's been the largest challenge that the program has given, to be able to, for the government to set up this infrastructure, which will then build for the next 20 years.
And we're very fortunate that that infrastructure is continuing to be built all the time. And you've talked about the importance of now trying to analyze some of that data and interpret it. Your group was awarded a Sarcoma UK Genomics grant in 2018, and you're also a lead for the Clinical Interpretation Partnership for Genomics England. Could you tell us a bit more about how that's going and why that's been important?
So the sarcoma team right around the country, Birmingham, Manchester, the London groups, were really active in recruiting patients to the 100,000 Genomes. So compared with many of the other much more common cancers, we have a huge repertoire of these data sets. And I think with what we've put together with the generous funding from Sarcoma UK, let's say it's like being a chef. You can't have a chef that just, well, if you go to a very mediocre restaurant, it may be very tasty, but if you go to a high-class restaurant, you'll have somebody who makes the soup, and you'll have somebody, a fish chef and a meat chef, and you'll have somebody who does your dessert, and you'll have all of these separate chefs. And that's what we've put together with the GSIP. We've somebody who focuses on the epigenetics, on the RNA sequencing, on the germline genetics to study the familial disease. So almost every component in the GSIP, we have brought in experts, and we're still open to bringing in new experts and people to collaborate with to make up those different components. So we have the best restaurant in the world. So
that's great, thank you. And just for people that don't understand the acronym,
that's
GSIP.
what we're delivering.
Sorry, Adrian, I think we lost you there slightly. I don't know if that was just me at the end. But for anyone who was wondering, GSIP is the Genomics England Clinical Interpretation Partnership.
So that is a bit of a mouthful, but essentially that the GSIP is the research group to analyze the data coming out of the 100,000 genomes study. And the government is also linked to that. It has to be developed a bit better, but with clinical outcome. And we're continuing to collect that clinical outcome from those 1,000 or so patients and add to it with the new genome medical service that's available for sarcoma patients now.
Great, thank you. And if I can go over to Zoe, so you're almost like a specialist restaurant in Adrienne's analogy there, in that you look at a specific type of sarcoma. Could you tell us a bit about the work you're doing with dedifferentiated liposarcoma?
Yeah, I was actually just thinking about what Adrienne was saying, and the restaurant analogy is a really interesting one, because I guess what we're trying to do with the specific dedifferentiated liposarcomas in this project is really create the perfect meal, as it were. So use all of those components that Adrienne mentioned, all of those different chefs, and understand how each of those components works together, and how we can then utilize that information to go back to talking about cancer, to understand what's going on in a patient's cancer. So traditionally, we tended to look at, say, single genes, single proteins, single elements in a particular cancer and say, can we find a drug to target that might treat the cancer? But what we're doing slightly differently now is saying, well, these things don't work in isolation. We know they don't. So how do they interact with each other? What makes them specific to both the individual patient, but the general patient, so the general patient population with dedifferentiated liposarcomas? Can we understand how those things interact with each other? Can we pinpoint key players in this network, that we call it, that will actually be more effective when we find treatments for them? So rather than just looking at an individual protein out of context, can we understand the whole cancer and how we can actually treat that better? And even better than that, can we then start to understand why people don't necessarily respond to treatments? Can we use this network that we've created, or all those components together, to really understand why, when treatments don't work, why they don't work, and how we can then find better treatments. And as you mentioned at the beginning, I'm really interested in finding, less harmful, more effective treatments. So it's about both of those components. Can we find something that works better than the traditional therapy?
Yeah, absolutely. And you've touched on to, how is this used in practice? We've got Andy and Alex with us, both of whom are treating patients rather than in the as well as being in the research side in Andy's case. And Alex, I don't know if you do any specific research, but Andy, Alex, how do you feel that genomics is changing the way that you're treating patients and the way that you're talking to them?
Well, I'll step in and answer if that's okay, Andy. I think the way that genomics currently informs the actual treatment of patients with sarcoma and how hopefully that role will grow is really answering two questions. First of all, just precisely what sort of cancer, what sort of sarcoma we're dealing with, and then what information is there to guide how best to treat this. For me as a medical oncologist, what drugs in the available repertoire are best to be reached for. And in terms of the actual diagnosis, telling us how precisely what kind of disease we're dealing with. That's really is embedded in current practice in terms of the sarcomas that are associated with very specific gene fusions, which tell us, for example, if we're dealing with a Ewing sarcoma or particular sort of rhabdomyosarcoma, to informing things like, are there drugs we can best use? And I think really the case of gastrointestinal stromal tumours, GIST, is really the archetypal example at the moment for a sort of sarcoma that, only going back perhaps slightly more than 20 years, was effectively untreatable in many cases. But then the discovery that a large majority of these tumours had particular driver mutations in a gene called KIT really revolutionized the treatment of that and led to the development of targeted treatments, first imatinib and then further treatments to now what is really quite a sophisticated genomics-driven treatment pathway. Now, so far, GIST has remained really out there on its own as an example of that sort of genomics-driven treatment. But what's really the hope now is that we can discover further examples by casting a broader net across the genome to discover these drivers and ultimately develop new treatments. And I think the discovery of Ntrack fusions is really a powerful example of that in more recent years. So Ntrack is a gene which can be abnormal really across a wide range of cancers, which happens in quite high frequencies in really quite rare childhood sarcomas, but we know also occurs across a wide range of other sarcomas in quite low frequency, which is really an example of how new discoveries can be made, which really kind of drive forward, first of all, telling us what sort of cancer this is and how best to treat it. And really another field of promise for genomics is another layer of information. So whilst Professor Flanagan can look down a microscope and tell me if there's a certain sort of sarcoma, if this is a leiomyosarcoma or a liposarcoma, what genomics is suggesting is going to show is that it's going to provide another level of categorization, which perhaps transcends the more traditional classification and really kind of moves us on to a new way of selecting different tumours for different sorts of treatment, which I think is really something that's showing great promise for the next 10 years or so.
Thanks. And Andy, you're used to, as a surgeon, removing sarcomas, as the first line of treatment for many, many types of sarcoma. How have you seen this impact your practice?
So I always joke in the lab, our lab motto is we want to get rid of surgeons because surgery, I imagine a lot of the audience, or a good proportion, will have been through surgery for sarcoma. And having done it myself and doing, it can be enormously disfiguring. Limbs can be lost, literally. We can do very large operations in the abdomen where we remove multiple organs. And so the whole point of genomics is if we can find what specific genetic change and mutation, as we call it, is driving a particular cancer, we can give a drug that will shrink it down. So we give it before we do the surgical treatment. The idea being we can preserve more normal anatomy. And as Alex has said, that's certainly the way it happens with GIST. There are other cancers where this is becoming more and more a possibility. So Professor Flanagan and others identified in chondrosarcoma. There's a change called IDH1 and 2. And there's a new drug out for that. That's in clinical trials at the moment, which has shown great promise. And that can be replicated across a lot of other sarcomas as well. So the advantage of these things, it's a bit like the way I describe it to patients, it's a bit like antibiotics. You send off your sample of your urine if you've got a weird infection, decide what it's sensitive to, and we can tell you what antibiotics to have. With genomics, we can tell you what drugs it might be sensitive to. That's not the be all and end all. The other thing my lab does is generate disease models. We take a sample of the cancer and grow it into a model in the laboratory and then expose it to the same drug to make sure the genetics is right. But from those things, they can give us clues towards how best to treat the cancer. So it's a very exciting time. The Ntrack fusion we've talked about already. We've just published a paper in the Journal of Pathology looking at well-differentiated liposarcoma and found some targets in the way that the body processes sugars, which might be targeted with drugs. So there are lots of opportunities that have only really come about through these radical advances in investigation.
Thanks, Andy. And you kind of both touched upon treatment choices there. I think it might be worth just quickly talking a bit more about the Ntrack fusions and larotrectinib and entrectinib, the two drugs that have been made available for that recently. Could you tell the audience a little bit more about kind of the future of medicine and these histology-independent therapies, as they're called, ones which target specific mutation in your tumor, and no matter what type of cancer you have, compared to what would be traditional treatment?
Sure. So just to go a bit deeper about Ntrack fusion. So again, just first of all, what are these things? These are effectively mutations across one of 3 genes that are called Ntrack 1 to 3, which have been found across a wide range of cancer sorts. And when these mutations are present, these gene fusions are thought to play a very important role in driving the cancer behavior. Now, really up until recently, the sorts of mutations and driver genes that have been looked at have really been restricted to particular cancer types. So there's a very well recognized repertoire of lung cancer driving mutations and then colorectal cancer driving mutations. But what's particularly interesting about Ntrack fusions is that these are mutations that seem to happen across different cancer types, but can still be targeted and treated successfully in the same way. And so that brought about quite a lot of excitement about a possible new treatment, which works very well for patients, including sarcoma. But we know from the available research that there are not very many patients out there where Ntrack fusions are driving their cancers outside of certain very rare subtypes. So a big challenge now is to know in whom to go looking for these mutations, and the goalposts in that are shifting. There was only within the last month a paper suggesting that in liposarcomas, which have already got a very well-defined genomic abnormality, which the dogma was, well, they're probably not going to be Ntrack fusions there, but what do you know? You can still find these fusions there. So we're still very much learning about where to go looking for these, but the main reason this is very much a worthwhile undertaking is that we know that if we find these gene fusions, that the chances of responding to these new drugs, larotrectinib and entrectinib, which really target the cancer drivers brought about by these fusions, is very high. So in some trials, about 2/3 of patients had considerable tumor shrinkage when treated for these, which lasted for months on end. So again, it's a very good example for the way that looking at tumors from a genomic point of view, rather than where in the body did these originate from, what do they look like under the microscope, saying, well, what do they look like at a genomic level, and that telling us about a way to best treat them.
Thank you. Richard, we haven't heard yet from you, so I'm going to bring you in here if that's okay. Sarcoma UK has committed over £750,000 to date for genomics research, with plans to reach £1 million by the end of this year. Why is it? I mean, we've heard a lot of very positive things, but in your own words, why is it that Sarcoma UK is so keen to support this research?
Well, I think the other panelists have highlighted exactly why, what we'd hoped to do and what we'd hoped would come out of it. I think sarcoma is a disease, a cancer that has been underfunded in the past. I think the genomics haven't been well understood. And surgery, as Alex and others Andy have mentioned, is a mainstay. It's the first port of call. So we wanted to kind of branch that out. So that's why we kind of 4 years ago set up the Sarcoma UK Genomics program. And I think we wanted to do 3 different things, and actually all 3 of the panelists have talked a little bit about this, but we wanted to bring together significant quantities of sarcoma-related samples, both tumours and healthy tissue, and their accompanying data from the 100,000 Genome Project that Adrienne mentioned. And we wanted to do it in a readily accessible format so that we could compare with other global databases, because sarcomas there are over 100 different types of sarcomas, and they're all relatively rare. We need to compare with other data across the world, so we needed to be in an accessible format. The second thing we wanted to do was compare these samples to identify genetic mutations that trigger the initial developments of sarcomas, so how sarcomas start. And then thirdly, we wanted to help us understand how tumours behave, how they grow over time, and what the various genetic changes that sarcomas undergo through that time that trigger growth. And by doing this, we hope that we give hope, I think, to patients and families, and make sure that they were so desperate to kind of see that hope, and hopefully the program has done that. And I think what we hoped has started to happen, and that is that it can hopefully lead to tests to diagnose certain cancers, but also more potential treatment so that patients can live longer.
And how have you've talked about some good developments that are starting to take shape. How have these new developments in genomics affected the wider work of Sarcoma UK?
Yeah, so although the work within Sarcoma UK is led out of our research team and research department, it has an impact across all parts of our work. So not only does it, the fundraisers, the communicators need to think about how we represent it to the public, because the public will only give us money for this type of work if they can see the benefits. But it's also on our information side. We need to do genomics information, whether that's about testing or whether it's patient information. It's impacted that side of our work. And then in your area, in policy and public affairs, it's impacted on the work that we do there. So what we lobby on, what we campaign on, and whether that's public campaigning or whether it's behind the scenes work, it has an impact on all aspects of our work.
Thank you. We've got some questions that I'm going to go over to now. Andy, I saw you were typing away on a question, but I wonder if we can bring that out into the chat. So, I mean, there's a wider question here from Dawn, which is around, are sarcomas hereditary and what testing can be done to find that out? Who would like to come in and talk a little bit about the hereditary links or not of sarcoma? Andy?
I can talk a bit about it because I run a family cancer clinic. In fact, I was running it this morning, so I feel like I can give some insight into this. So to answer your question, first of all, Dawn, about Li-Fraumeni, obviously, Li-Fraumeni is one of those conditions that a lot of people heard about because it's associated with sarcomas, and it's devastating because it affects people at a young age. And it's because of a fault in a gene called TP53, which is, it guards your genome, it keeps it stable, it keeps it healthy. And people who have Li-Fraumeni have a fault in it, and it can lead to them getting things like osteosarcoma. Now, people generally tend to develop that when they have Li-Fraumeni in their teenage years, although it can develop later. But there are other diseases that also cause types of sarcomas. There's a type of tumor called a desmoid, which is benign, but become malignant, that is associated with a disease called FAP. There are some other rarer sarcomas that can be caused by mutations that are hereditary, although it's relatively rare compared to some other cancers. My advice for people who are worried that they might have a family history is to get referral to your local clinical genetics unit. There's one in each region in the UK, and all you have to do is get your GP to refer you along, and you'll go and see a clinical geneticist, and they'll go over your family history. And if they think it's appropriate, they'll arrange you to have testing. And then generally, if people are found to have a mutation, they then come and see somebody like me for risk management. So there are many, once we identify people, we can usually take steps to try and reduce the risk of developing a cancer in the future.
And more generally on the sarcoma side, Andy, how common is it that there's a hereditary link? Are there any other proven links apart from this particular syndrome, or would you say that there's not such a hereditary side to sarcoma as it is?
There are some sarcomas where being hereditary is important, but if you compare it to, say, breast cancer, where 10% of women with breast cancer will have a mutation in the BRCA gene, it's less common. So the soft tissue sarcomas that I see clinically in the family history clinic, they rarely have a germline mutation, but there are some specific diseases like osteosarcoma where you must think of it. I think Adrian has got a point she wants to make about that, because she's the expert on osteosarcoma, not me.
Adrian, you got your hand up.
Well, I know I absolutely agree everything that Andy has said. I think the osteosarcoma is one, if you're younger, up to the age of 30, I mean, the data now is showing that up to 20% of patients with that particular disease could have a germline alteration. So I think it's important. And I think one of the reports that will come out, an important finding from the GSIP and the 100,000 genomes, by pooling the sarcoma data and all the other cancer data, we will start to provide really useful data on familial disease in sarcoma in a really powerful way. It's going to take a little bit longer, but I think it's something that the GSIP and the funding that Sarcoma UK has provided will produce.
Thank you. We've got a question from Sheila, which is about her daughters not being eligible for chemotherapy due to low white cell count. I think if I can broaden this out very slightly, are genomics allowing us to change the treatment pathway for people who are not able to have chemotherapy, or does this something further down the line? I don't know who'd like to come in, Alex, perhaps.
I think that's a really interesting question. I mean, Sheila, just to directly ask you questions, it's tricky for us to comment on that directly because there's so many kind of details that are important. But I'd certainly suggest you speak with your daughter's oncology team about that. They'll be able to answer both about chemotherapy and what level the white cells need to be, but also for clinical trials as well, where these things are often kind of dictated as to what level they need to be. Brad, just in terms of the broader question about how genomics can inform selection of patients, I think that's a whole other field as well, because we're talking about a lot about what the tumor genomics are, but then kind of touching on about germline genomics, how that might inform why the tumor occurred in the first place. But of course, the genes that don't have anything directly to do with cancer can play a big importance in terms of how our bodies metabolize chemotherapy drugs, how we react to the various stresses that cancers put under, how we react to radiotherapy. And at the moment, there's not really very much recognized as being consistently important for that. There's certain inherited conditions which certainly make things like radiotherapy a bit more risky, and the risk and chances of reacting to that. There's certain chemotherapy drugs where it's recognized that if you have a common polymorphism in a drug called DYPD, that your risk of serious side effects is much better. So you're best off avoiding that. But really, these are just a small handful of examples that inform us as to how we use, say, chemotherapy drugs. And certainly it's a field where we can learn a lot more about how specific germline effects inform how individuals handle different chemotherapy drugs, and then which could potentially inform about what drugs to use in the first place, how to optimally dose patients, how to best treat side effects. So I think that's a really important area to keep in mind about how genomics might be able to help.
Thank you. And Andy, you'd like to come in here as well.
Yeah, I just remembered an interesting paper that I think will hopefully put people, it sort of helps some people and doesn't help others. It's about whether you inherit sarcoma. So this is all public knowledge. You won't mind me telling you that the head of commercial partnerships for Genomic England is a man called Parker Moss. And his daughter rather sadly died of neuroblastoma at the age of 7. But because he was very generous, he contributed to a tumor for whole genome sequencing. And what they actually found through something called ancestral study of tumors, they found that her tumor started when she was in an embryo, is when her cells were divided at the 20 cell stage. And so it wasn't something she inherited, but she got it very young, was because it happened as a random fault way back when she was in the womb. And it was nothing anybody did, and it was nobody's fault. It was unfortunately just bad luck that this cancer developed. It does answer some of the questions now about why some people develop cancer so early. Sometimes, and most of the time, it's not just because it's something you inherit. It's a lot of the time, unfortunately, it's just because of bad DNA repair and bad luck. And it's unusual to have something like that that's passed down through a family.
Thank you. Some of the ways that we're going to use this information is through trials. Once we've kind of done all of the interpretation and we have a theory. Pauline's asked, how does someone get onto a trial? A very simple but important question.
I'm happy to step in that. So clinical trials are a very varied beast. So there's lots of different trials at different phases, focusing sometimes very narrowly on particularly patients, sometimes much broader in their focus. Because of the rarity, trials particularly focused on sarcoma don't come round too often compared to other cancer trials, but there certainly are those out there within the UK and also further afield. So part of the challenge of getting onto trials, first of all, knowing where there are trials and where they're open, because trials are always very specific to site. So a trial that's open in Edinburgh wouldn't necessarily be available in Exeter, for example. And there's various resources you can access that, and certainly Sarcoma UK is a good resource for accessing that. And I know that the team there keep in view about all the trials that are out there. In terms of then getting on a trial, it's just a case of asking for a referral. your team will have some sense of whether or not you're a fit for that trial. And if they feel they are, they'll be able to refer you on to the centre for consideration of that. Trial participation is always subject to meeting quite strict eligibility criteria. So it's not always the case that all patients can get onto all studies. But certainly, as someone who runs clinical trials at the Christie, I'm always very, very keen to try and offer patients trials where possible, and certainly they offer opportunity to potentially get access to treatments that may prove to be helpful. So, in short, kind of speak with your medical team, speak with Sarcoma UK to see if there are trials out there that might be a fit.
Thank you, and we'll put the information for the support line in the chat for the way you can contact our team of trained specialist nurses to talk more about different trials and options, or anything at all that you have to do with your sarcoma. Roger has asked about when is it best to use genomics to impact treatment. He says, is the most valuable potential for genomics when we can bring the analysis closer to surgery to try and prevent recurrence? And linked to that, if we only use the technology to chase recurrent cancer, we won't make a big enough difference in survival. I don't know who would like to comment. We won't make a big enough difference.
I can hopefully answer it, but go first, sorry. Yeah, you go first, sorry.
Yeah. I was going to say, from a lab perspective, this is where models are really important, and this is where genomics is really important. So not only understanding what's going on in any given cancer, but trying to understand the difference between a patient who will recur and the wealth of genomic information that we have will allow us to do that better. And the models that Andy mentioned means that we can really start to understand which patients might recur, why they're recurring, and what their genomics has to do with that. And then we can try and head that off at the pass. So we can basically say, okay, we think these patients with these genomic changes are those who are most likely to recur. Therefore, we can then front load, those treatment decisions and things like that. So, the models, all the information we get through the GSIP and everything is really key to understanding that process and exactly head off the pass, rather than just trying to treat those that have the same genetic information.
Thank you. Andy, you wanted to comment on that as well?
So it's a good point about trying to get it for its recurrence. That's exactly what we're trying to do. We're in negotiations with a drug company at the moment about hitting people with chemotherapy for their sarcoma before they have an operation. The idea to make it less likely to come back. But the way we're doing that is we're going to undertake whole genome sequencing of the tumor before it gets to surgery. The idea being we've got a panel of drugs we know do work against sarcoma, and some ones that we think might. And as Alex has said, there are certain changes that may be possible against sarcoma. So we want to make a kind of study called a basket study, where we choose the right patient for the right drug, and then we give it to them before their surgery in an attempt not just to shrink it down, but to prevent it from coming back in the future. And that's where I see genomics going. That's being replicated across other cancer types. So it's a bit further along, but if you look at breast cancer now, if you have a type of breast cancer called a triple negative breast cancer, a lot of women now have chemotherapy before surgery. And in highly targeted triple therapy, 20% of patients, their tumor shrinks away and goes completely. So it's completely transformed it. And that's where we want to go with sarcoma. We want to try and find the right genetic alterations, either shrink down or prevent recurrence using sequencing to guide us, and models like Zoe talked about, and then move it out into surgery that's less horribly invasive than previously.
Great, thank you. We've got a question from DT, and we've touched on this slightly already with Alex, but she says, is GIST sarcoma the same as other sarcoma development? And I think we're coming at this from a genomic angle. I have silence. Would anyone like to comment on that?
I could talk about the molecular genetics of GIST until you're blue in the face if you want, but the answer is one of the distinguishing things about sarcomas is they're basically all diseases of the connective tissues of the body. But what's different about GIST is the gene fault in it, as Alex said earlier, is very well known, and that's why it's better studied than other cancers, because we have drugs like imatinib and a couple other drugs that are targeted against GIST. So we're a lot further ahead. It is a type of sarcoma, but we have better options than we did previously, and a lot of other my centre and other centres regularly treat GIST tumours. So of all the sarcomas that are around, it's the one that's probably the best studied.
Thank you. And then I've got a last question that I'd like to ask everyone, if that's okay, and that's, what are you most excited about in genomics for sarcoma? Zoe, we'll come to you first, if that's okay.
For me, it's all of that information that we have available to us now, thanks mostly, to the fact that patients are so willing to engage with us, and that we can really understand, how cancers arise, where they arise, why they behave the way that they do, and genomics has just made the whole thing sort of explode with information that we can really use to drive better treatments. And for me, it's about, can we repurpose some of the drugs that are already out there so we can try and find better, more effective, less harmful treatments and get them into clinical use as quickly as possible, and work so closely with the surgeons to get from the patient to really understanding the genomics to this kind of precision medicine, really targeting the genomics to treat a patient. And that's just so exciting.
Thank you. Alex, would you like to go next?
I think it's really going to be the rollout of the availability of a much more kind of detailed genomic assessment of patients with sarcomas tumours. And this is really the legacy of Adrian and others' work with the 100,000 Genome Project. So really, as of now, there is availability to be doing whole genome sequencing on sarcoma tumours, which is still in the process of being incorporated into everyday practice. But it really provides the potential to generate enormously detailed genomic information about ultimately every patient with sarcoma. And the power of that is it really does allow us to kind of drill down and ask all sorts of questions, many of which we've touched upon this evening, about why has this sarcoma happened? Is there a hereditary basis? Does this allow us to classify with other sorts of sarcomas with similar genomic basis? How can we best treat this? Is there something about the germline basis which tells us that to avoid this treatment because it's going to be too toxic, whereas this one might work better? So really, it's going to be a tremendous resource which we're going to continue to learn from, for really certainly the full span of my career, and then likely beyond, which, today now, if I'm presented with a whole tumor genome for many of my patients, I know that it's going to be the rare occasion in 2021 that's going to tell me precisely what to do. But hopefully the day where it's not too far away, where that's going to really make a big difference for our patients.
Adrian, as you were name checked there, can we come to you? You're on mute, Adrian.
Well spotted. So, well, I agree with everything that Lee and Zoe have said. So for me, genomics is at the heart of understanding the whole of cancer. And if we're studying sarcoma, it's the heart of understanding every single aspect of sarcoma. Diagnosis, giving patients prognoses, that's very valuable. What their outcome is going to be, how they plan their lives. If they're given therapy, are they going to respond to therapy? Are they not? Can we get new drugs? Can we develop new ctDNA acid liquid biopsies? I think it's just genomics is at the heart of this. And one of the things that excites me about from coming out of the 100,000 genomes is I think it's brought us together as a community. There's so many things to work on. There's all these different people around the table. I think we can share benefit. We can work together. We can add value for patient benefit. I think that it's brought us together. It's making us a much more powerful strength. And one of the things that has really made a big difference, I think, from the 100,000 genomes leading into genomics is that it has recruited a good number of younger PhDs, medics and non-medics, into studying sarcoma. So, I see a raft of pathologists and oncologists now coming through who are really going to be the next generation. They'll be really on top of this discipline. And that's 10 years ago, that just wasn't there. And the 100,000 genomes and the genomics world has transformed that. And I also think we should thank Sarcoma UK for, investing so much into this and trusting us to do this work, and to thank the patients for giving us their samples and trusting with their samples and engaging with us, because without them, without you, it's just not going to happen. Yeah.
Really, thank you. Andy?
I completely agree. Once again, with everything everybody said, it's only thanks to Sarcoma UK that work in my lab would be funded. I'm a bit of a geek, so I love cool technologies. So I think for me, the most exciting thing there is one of the things we've always struggled to understand is why people's cancers become resistant to treatment. And that's because tumors evolve like bacteria, like COVID. I'm afraid to say this, drop the C word into this, but the cancers evolve when they're on treatment. So one of the new technologies we've got is we can disassemble a cancer into its single cells and analyze it. And Sarcoma UK are funding us to do that on a wide range of sarcomas. And that technology has already given us insights into liposarcoma. We've already done quite a few tumors for it. And it's going to give us really, really big insights. It's going to be the next leap into understanding how we can better improve cancer treatment. And so for me, that's the most exciting thing at the moment.
Great, thank you. And last but not least, Richard, would you like to wind up for us? You're on mute, Richard. Sorry.
I was thinking as the others were talking that there's been lots of bad news in sarcoma. There's not much good news, but actually, I think what genomics has given is promise, and I think that's the beauty of it. And just some of the questions in the chat have been really sad and tragic. Stories of how it's impacted on patients and continues to impact on patients. So hopefully, I think genomics can lead to tests to diagnose cancer earlier and potentially more personalised treatments that the others have said. So that's the hope for me for genomics. And again, the others have said it, but it wouldn't be possible without the support of patients, whether that's patients giving their samples or permission to give their samples, or for people giving money to organisations like Sarcoma UK. So it's got an important part to play.
Thank you very much. Well, that's all we've got time for. If your question wasn't answered, please do get in contact with our support line. So that's sarcoma.org.uk/support line, where you can find their information pretty prominently on our website and in the chat box. I'd like to extend an enormous thank you to all of our experts for being here and giving such fantastic answers, and also to everyone for joining. And with that, I'm going to pass you back over to Enda. Thank you.
Thank you very much, Bradley, and thank you to everyone for such a really illuminating discussion, I think, about genomics, and I think a source of great optimism, I think. I'd like to thank the panel particularly for making it understandable to the lay person such as myself. I hope that's shared by the people who attended as well. So a recording of the session will be available tomorrow, and we'll post links to that on our social media and also at sarcoma.org.uk/Zoom. Try saying that quickly. Also go to our sarcoma.org.uk/10 years for a list of all the activity taking place around Sarcoma Awareness Month and our 10-year anniversary. So finally, let me thank the superb panel for sharing their knowledge and experience tonight. So Professor Andy Beggs, Richard Davidson, Professor Adrian Flanagan, Dr. Alexander Lee, and Zoe Walters, thank you so much for your time. Thank you also to Bradley for hosting, and thanks to all of you for coming along and taking part. We have 2 webinars next week, which is the final week of Sarcoma Awareness Month. The first one is at the same time on Tuesday, Living Well with and Beyond Sarcoma. And on Thursday, we have a session called Supporting Someone with Sarcoma. And it's aimed at families, friends, loved ones of people with sarcoma who maybe haven't had a chance to get up to speed with the various things that sarcoma brings up because of the pandemic, essentially. So if you want to tell your loved ones to join us on Thursday at 7 p.m, please do. They do need to register, but it's free. And the details again are at sarcoma.org.uk/Zoom. So just finally, again, thank you to the panel and thank you to you for arriving tonight. Have a lovely evening.